So I'm Jay Etchings, Director of Research Computing from Arizona State University. I'm sure many of you had been at the keynote this morning where we kind of discussed part of the global vision and what we're doing in population genetics and human genomics at the university. My department does the computation and data-intensive work for the whole university. And as you know, Arizona State University is a Research One university with the largest enrollment in the country and I believe largest square footage in campus too. So we are the new American university. I did skip that slide that talks about how we're number one, although I think it is important to say that we are number 4 in medical research, public funding opportunities in schools that do not have a medical school. So we're very focused on partnerships with and external relationships where they can bring us things that we don't have, like clinical data and of course, public sources. So here's some other interesting factoids about the university. We just got these no walk zones last year, or no ride zones, or walk-only zones, whatever you want to call them. They're great because there's 13,750 bicycles on campus every day, never mind the hoverboards and skateboards and you name it. I think the bicycles propose more of a danger than the 19 species and subspecies of rattlesnakes we have on campus as well. So when I took the appointment to the role as Director of Research Computing, we had an existing infrastructure. We brought in a new hybridized cloud infrastructure and data-intensive components. And then we had the clusters, the closet clusters we like to call them, that existed around the university that we needed to bring in and aggregate. And so that's kind of the 3C model we adopted here, where we took a large amount of traditional computation and sent it up to the cloud. We use the Penguin on Demand service. We run all of our data-intensive workloads local on a large-scale cluster known as the NGCC. And then we also maintain the university-owned condo structure. Anybody familiar with that term, condo? Right? Okay, yeah. Condos where we have a large converged cluster made of many nodes. People invest in it. It's like a timeshare, but for compute. Right? So we are open source focused. And ideally, we take components that existed in traditional HPC and find their counterpart within the Hadoop ecosystem and then right size and move applications over. And we did this based on some consultation we had within several different teams to look at some of the things that were being done at Google, find the open source equivalents, and then build there. I guess I'll say probably the piece I didn't say this morning is the emphasis on open source is key to us, because as somebody who sat as a panel reviewer at the National Science Foundation, if you look at some of the structure of the proposals now, if you're going to build something with public funding and public money and it's going to remain proprietary, you have to write an addendum to put that in there. And I don't know if that in any way impacts your competitiveness in that proposal, but it would be my thought that if you're going to develop something with public money, chances are you're better off to develop something that then will be put back into the open source market. All right, so we had talked about this morning. So Dr. Ken Buteau's vision in coming from the NIH and working on the Cancer Cloud project was around the transcore knowledge framework. All right, so it's essentially a seamless fabric where the traditional components that we had in transactional and in large-scale HPC, in Internet 2 connectivity, and open big data are all on the same fabric without a need for ETL or other mechanisms, and where we can track everything from file provenance to metadata throughout the infrastructure. So our hybridized cloud model from 2012 looked like this. And I keep this slide because it still looks a lot like this, which I think is a testament to how forward-thinking this was back in 2012, that many of these components still exist. So the human genome being unique data, Some folks will tell you it is, some folks will tell you it isn't, although I thought this was neat from the TED Talk where they showed if you'd printed it out, essentially on 8 by 11 sheets, it would be as high as the Washington Monument. So this was kind of a cool piece here. But when you hear these talks, and you see that other slide, that, and I didn't bring it today, I probably should have, but it's in every single, if you go to BioIT World, you go to any conference where they talk about genetics and genomics, you see this slide that shows the skyrocketing output of genomic data and the drop of the price, right? Everybody's seen that, right? Now we can sequence a genome for $1,000, right? Well, that is true. We can, but that has nothing to do with the analysis. And I think that gets overlooked quite a bit. There's nothing to do with the analysis. You essentially sequence it and produce the data, and that's where you start. So we've at least reduced the hurdle to the start of what is interesting science from what used to be millions of dollars down to $1,000. Also, when we look at the genome being 3.2 times 10 to the 9th, well, it's actually a little bit bigger than that, right? There's 3 billion diploid base pairs, 6 billion haploid sequences, and that's important to know, right? Half from mom, half from dad, and understanding that really has an impact on the science. And then, of course, the limitations that we talked about before that we suffered both in HPC traditionally and in MapReduce when we did them, right? The impact of running these jobs, even though in greater parallelization, but via a batch job, was inefficient. Just took too much time to do it. And then, of course, I think everybody's aware of the standardization gap within academia, right? It's not so much in the enterprise as it is within research institutions, because as a grad student, you're going to use the stuff that you want to use. You may even build your own file system. It sounds crazy, but we've seen it before. They're out there. And they don't necessarily play to any sort of standardization or sustainability for the future in this science. So I stole one of Ken's slides here because really for us, we don't want to focus on big data, right? We want to focus on big answers. I had talked to some of the folks out here in the vendor area and talked to them about just genomic data in itself, how its extreme longitudinal nature of the data makes it very unique to be 3.2 billion characters across and only 2 deep. Is that big data? I don't know. But we can get big answers from it. Right, so if we look at tumors, it was only a few years back, it seems, that we realized that tumors, even though the cells look like a big mess when we actually look at them, were actually unique, identifiable cells at one point, and that there's heterogeneity, right? So in knowing that, it's helpful to be able to understand the evolution of a tumor by understanding the cell makeup and then run simulations based on that. So the simulations that are interesting, and these are the science questions, and I'm thankful that I have Dr. Ken Buteau sitting in the front row here. This work, this work that's done on this tumor simulation mechanism was done by Dr. Carlo Mele, Dr. Wilson Sayers, and Diego Chal at ASU. Those are the scientists that have put this together. And it followed much of the same evolution of how we did one of Ken's early NGCC projects, where we took it from traditional HPC, we took it from doing things in Plink, we put it into something called AmpLab, the pipeline out of UC Berkeley. Is anybody familiar with that? It was a cool set of tools. It was ready to go in Spark. We still use some components of it, though, although I think it's kind of exhausted its funding life cycle. And it may actually become a piece of proprietary software. I don't know. If anybody knows the history or where it's going next, come and tell me, because we had imagined that years down the road that it would become some sort of a Cloudera product or something like this, just because of that close relationship and Hammerbacher's influence. But anyhow, in running multiple jobs and simulations like this, we had actually at one time, well, going back about 2 years now, tested all 3 major distributions. And we had tested them against traditional HPC and produced a whole bunch of metrics to try to figure out how we could do this the most efficiently. And we came out with a winner head and shoulders above the rest, but then we also had to factor in other parameters. And those were simple things like the alignment with open source and being able to return these components back to the community, because that's one of the things that we do like about the AmpLab project. So in looking at this problem in itself and kind of understanding the exponential math behind this and the amount of simulations that need to be run to get the results that we're looking for and achieve the generation time. And I know it says down here it has an average of 4 days. Originally, when these were run, and this will sound crazy, but they were for single tumors with minimal variations. We were somewhere in the realm of 100 days. That's a real number, right? And at one point, as we moved it into MapReduce and better parallelized it, optimized some of our infrastructure, put it on newer infrastructure, we actually got it down to the point where it was closer to 60. However, when we get to the next slide here and we show you exactly how many simulations had to run, you'll realize why that wasn't sustainable either. So here's kind of the simple piece that I put together so I could understand it, right? So if you look at this, there's really 3 outcomes here. So the process starts with a single cell, then there's a driver mutation, and there are passenger mutations and driver mutations. Driver mutations is the one we're concerned about. The first possible outcome could be death, division, and a driver mutation for each particular cell. So that turns out to look something like this. And if you look at this in the natural setting, what you realize is it could take years for this to happen, but not in every case. So the next interesting science question is, well, why? Right? So if you look at that first outcome, 98% of these mutant clones die out early. Some of the tumors develop very quickly, and they appear to mimic other childhood cancers. Some take decades to develop and mimic adult cancers like melanoma. These particular tumors that are looked at in this study too are colorectal and glioblastoma multiforme, if anybody's wondering. So still, computationally very intensive, right? Originally running 10 to the 9 cells is not efficient on a laptop, nor is it efficient on your closet cluster. Having tumors die out before reaching a detectable limit or exhausting the run cycle doesn't produce an actionable limit. So we need to reduce the time, the run time, track all the mutations in the subclone size, and that needs to be reduced massively, right? So we had some basis to do this. We had some experience doing this because under the MGCC project, we had looked at doing EQTL analysis, right? So, so in brief, expression quantitative trait loci is the way we track regions in genomes containing DNA sequences that influence expression levels in one or more genes. And we had looked at building a pipeline to do this, to essentially put this in what would be a web-driven format, because it's a complex computational algorithm to arrive at. However, it's common enough that it should be available to a multitude of different researchers. So here's an old slide before that shows, what we're doing in HPC as far as times. Now, I guess an important takeaway from this slide is every one of these will run on the exact same hardware. This was E5 2650V2 processors. I think they had 128 gigs of RAM. They're Dell R720 XDs with 22 terabytes of disk space, 10 gigabit drive, Ethernet adapters. So all the exact same machines. And to assure that, we built these cluster pods and then wiped them and put the other one on. I guess if you if you decide to follow me on Twitter or send me a nastygram or buy my book, I will give you the slide that shows all the tests we did against all the top Hadoop distributors and what their outcome was. And we can do that because we're in academia. So, but because that's an interesting factoid as well. But one of the other things that we arrived at in doing this was one of the pieces that didn't get tracked. And when you look at great work that's being done in the national labs, they have a lot of maturity in the applications and scripts and routines that they've built. In academia, we don't. We have a certain subset of tools, then we build new tools. So we had found out that we had a couple researchers that were building scripts that would go through and parse the data at certain points, so then we could go ahead and crunch the numbers, right? That's wall clock time. We weren't even counting wall clock time. So when we finally get back to figuring out, well, geez, how long did it take to do that? And for each one of these data sets, how long does it take to do each individual one? And the guy we had doing it, it's actually a really good programmer, a really good scripter. It was still taking him a couple hours for each sample sometimes. So he would do this for an hour or two a week. Well, that fits in here as well, and it's an important thing to track. One of the cool things about Hadoop that we found out right away was this data was automatically parallelized, right? It didn't have to be built into an MPI job. We could automatically do that, right? If anything, the only deficiency early on we had was managing the size of RDDs and making sure that we didn't eat up all the memory that was required for the operating system on the individual nodes. And we went through a process to systematically upgrade RAM about 3 times in a 6-month period to continually put more in. I wish we had known that. That's a great lesson learned. I wish we had known that when we had started, but we didn't plan that way, right? So at Spark, took a day to get a tumor in a 10 to the 7th, still 2 orders of magnitude too small, right, because we said 10 to the 9th. We had existing code that was in MATLAB that needed to be converted to Scala. And I also have some information here that I can share if we, during the Q&A, where if you're interested in other pieces that were used, whether it's some of the R mechanisms and other things we pulled data with, I didn't put them in here, but I can share those as well. However, we had to convert the code over to Scala. We had an engineer that we hired from Early Warning that came into our team and joined the university after one of Ken Buteau's talks, where he gets to the end and he says, hey, we need people to come and join our team, and he did. And he, on the job, learned Scala and managed to get a lot of the stuff cut over. So now it takes seconds to simulate a single tumor, right? So now we can generate tens of thousands of possible tumors, thousands of outcomes. All right, so the standard output we would get looked like this. This is, and I have one more slide like this that will show the tumor outcomes and standard outputs. And I guess what I would say about this, because this is typically what you see in an academic paper, right? This is what you're used to, or what you'd... So who in the room does this not really have much meaning to? Anybody, or does everybody get it? Okay, no, be honest, right? So that's a problem. That's a problem, right? So we were lucky enough that the university had an academic relationship with Tableau, because great set of tools allowed us to take this data and put it in this format. So now we can tell, based on the driver mutations we produce, we can look at the cellular fitness and determine not only the growth rate, but also the amount of mutations that are created from each one of the cellular clusters, but also the ones that would die off sooner. So this is great when looking at the emerging world of pharmacogenomics and targeted therapies, because now we can start to look and figure out, okay, which therapies and specifically which pharmaceutical therapies and remediations target which cell types and which ones will be successful. So if you look, and there's a great book and documentary about the history of cancer by, help me out here, I'm trying to think of the gentleman's name. Mukherjee, right? Yeah. And, you know, when you listen to him talk and you hear the history, what you'll see is we've had chemotherapy treatments around now for, 60 years, maybe longer. And although in the past 60 years, the ones where we've come up with the neat acronyms for which ones are involved, and it's typically the prednisones. And what you see is a medication being delivered based on a diagnosis that comes from the standard playbook, and that's really what precision medicine addresses. The caregiver or providers can now start to work with a targeted therapy based on your specific tumor. So, and somebody said it this morning, every cancer per person is unique. It's not the same. Even if we both have glioblastoma, that's probably a bad example. So let's say colorectal cancer or hepatocellular cancer, it's unique in some senses based on the cluster of cells that are impacted, right, and how it grows. And that would be no more better demonstrated by the amount of people. I would say everybody in this room, by a factor of 1, is impacted by cancer, right? Is there anybody that doesn't know anybody that's had cancer or had an experience? But they all have different stories, right? They all have different experiences, right? You hear about Ted's wife who, survived breast cancer, and then you hear about, Jane that you worked with who died after 3 years of treatment, right? Well, why does that happen? Well, this is precisely why it happens, because we don't have precision interventions that address the patient. We do them from a broad spectrum, right? So if you look here, you can see that just this drug treatment alone, and of course this one was one of the ones that was sent over to me, and it actually kind of shows mutations that are resistant to treatment and then some remission. However, when you kind of look at the evolution here and how this ends up, it still looks like a pretty efficient treatment, even though there is remission in this particular case, right? And there is resistance to the drug. However, the result is pretty positive, right? It's pretty favorable. So if we go back to that original first slide we're looking at for the first tumor type we're looking at, right? We can see here, and this is kind of the final output. So it essentially has the population dynamic on it, then it has the subclonal computation, and then of course measured by the cellular fitness based on the driver mutations produced, you get this kind of full view of how this treatment plan should be laid out. And you also get to get a good understanding of the cellular makeup of each one of these particular tumors. And here's number 2. Very different, right? So it is fascinating that 2 of what is said to be the same tumor, same type of cancer, has 2 vastly different types of subclonal composition and populations of cancer cells, right? So you go back here, kind of different, right? A little bit. Well, that's important because one patient A may respond to the treatment, patient B may not, or may respond very quickly and then suffer relapse within a very short period of time. So, conclusions. So, I think there's a great argument here that goes in the realm of precision medicine to know that reducing time to research, being able to run as many simulations as we possibly can, and this is nothing new. It's nothing new to biology. I don't think it's anything new to computational sciences. I think anybody would tell you that folks at the labs try to run as many climate simulations as they possibly can over and over and over. We were talking earlier today at the Cube interview about agent-based modeling and computational archaeology and anthropology to determine everything from behavioral statistics. It comes down to the ability for us to run multiple simulations and get to that result as quickly as we possibly can. And that's how we can kind of start to build these targeted therapies and have faith in them to start to evolve things like and determine where there's drug resistance. So, as I had said before, here's the brilliant people that worked on this project. It's my buddy James Napier that we hired there. He's on Ken's team. Diego, Melissa Wilson-Sayers. All this work was done at ASU in our Research Computing Center, right down here at M. Wilson-Sayers. You should probably follow her on Twitter, because as this work goes forward and as it's getting to the point now where it's ready for further publication, there's definitely some interesting science here and interesting overlap with existing work that we're doing. And I believe I've made these available to the organizer anyway, so you'll be able to go down and get them. Here's our book, Strategy of Biomedical Data Science. Not necessarily a biology book, not really a infrastructure book, a translational text that talks about how biomedicine and computer science can come together, and this can give you an understanding of exactly how they fit together and hopefully generate, at the very least, some interesting conversation around how we're going to continue to meet the challenge of precision medicine, how we're going to respond with the appropriate infrastructures and software development to meet the challenge that we now have. So, well, that's my piece, and I got a couple minutes.
Yeah, quick question here.
Yes.
Is the assumption that all the research which you kind of did using Hadoop in this domain is parallelizable? Because if it's not parallelizable, it's not kind of Hadoopable, right? I mean, because I've faced several problems in things like expectation maximization algorithms, which are not necessarily parallel, So Hadoop doesn't help. So I'm assuming that all of this in the genomic domain is parallelizable.
I wouldn't say all of it, but, and granted, for every one of these cases, there's an outlier where you're going to say this doesn't necessarily fit. And this is the primary concern we had with traditional HPC. In some of the large organizations, other research institutions we worked with, they had a really good hammer, and therefore everything looked like a nail. And so your statement is spot on in the fact that not all of these workloads are parallelizable. However, the ones that involve this type of computation, especially where we're talking and taking a multitude of different tumors, different tumor types from a large cohort and then applying a whole bunch of variations to them, ideally it's a very highly parallelizable workload, and it benefits us to run that as many times as we can in the most, if you said to me tomorrow, hey, I'm going to give you another 1,000 nodes, that would be great, because we could be able to parallelize across that. So yes. I don't even know who to go with here. Who raised their hand first?
Why do you need an extra 1,000 nodes? Can you do something like SETI at home, Einstein at home, some project that?
Oh, okay. So a good example is Stanford's Folding at Home. It's a protein folding site that you can use. Certainly we could. Certainly we could use, and so the vision of the NGCC is one that is of the hybridized cloud. So if we could find other research partners to work with, and based on recent news, maybe it's Lawrence Livermore, one of the other labs, where we could share some of these workloads across. Yes, I will never come out and tell you that I want to buy infrastructure so I don't really want to run a data center. I want to do interesting biology and interesting science. I don't want to run a data center. So if we could put those workloads there, 100%, yeah, we would do that. Unfortunately, at this point, if you look at some of the offerings that are out there, whether it's Amazon or others, it takes a lot of configuration at the node level for us to fire these up, and they run a long time. And then the other deficiency is there's a lot of data that goes back and forth. So I think until the public cloud really redefines its profit model, I'd love to see multiple institutions like us peer together to do something like that. So yeah, perfect sense. Rather than buy 100,000 nodes, I'd rather use nodes all around the world. So yes.
So from all your research, would you say there is any correlation to genetic predisposition for cancer, or is it more towards lifestyle, food intake, and all that?
All right, Ken, that's you.
One of my ex-mentors, Joe Pompeo, he
strong evidence that actually it's almost assuredly code that we actually have. There's cancer that has very strong predisposition. There's cancer that has genetic susceptibility to environmental exposures, and then of course there's cancer that if you smoke enough cigarettes and live enough dissipative lifestyle, you can overrun the best of your genes. So.
Anybody else? Oh, yes, sir. Well,
You said you had to increase the memory threefolds. What kind of machines do you run on?
oh, am I there? So at the time, we had the original NDCC was built in a kind of a public funding opportunity where we purchased somewhere around 44 Hadoop nodes that were Dell R720 XDs. We bought them because we, at the time, were more concerned with the big data and not the big questions and big answers, and that's why I have that slide in there. So we wanted to jam a bunch of disks in there. I think we started out with 64 gigs of RAM each, and then we went to 128, and then we went to 256. So we just, as we brought in the workload, I don't think we were able originally to extrapolate how big that would be because, you know, the wonderful thing about working with research scientists are many of them have no idea how computation works. And because of that, it's like, well, it's kind of like how honest your child can be sometimes. So they say, wow, this is fantastic. You can do 10 of these? Can we do 10 million? And you go, well, I think we can. And that's when you have to buy more infrastructure. So that's the path we went down. In hindsight, if I could look back, that's a place where, because we were early adopters of Spark, at least in biomedical informatics, it would have been a good lesson learned. Yes?
So among your slides, I saw a pretty dramatic advantage, say, for Spark compared to MapReduce. But I guess the MapReduce you run was without tests. You know tests, right?
Yes.
Yes.
Yep.
I wonder if, of course, this is up to your discretion whether you have the resources to try out MapReduce on this.
Well, you know, and we had kind of gone down the path with a couple other groups in our computer science department to look at to both look at streaming, to both look at a couple different ways to improve the MapReduce performance. We even built machines that had greater volumes of SSD on them. At this point, I don't know, and I'd love to hear your ideas. You can send me an email and let me know what you think. We'll share some data with you to figure it out, because if there was a way to do that, we could maximize our utilization across some of the other clusters we have that probably don't have the appropriate memory pools to run Spark the way we have been. So it's definitely an interesting topic, but we tried Storm. We tried a couple different approaches. Originally at one point in one of our POCs, we were running MapR, and it was the file system was considerably faster, right? Their version of Drill was actually really useful for most of our researchers. However, it was still tough to build into a funding opportunity because I can't then build something and hand it to you, and you'd say, well, I have to buy licensing, and that's kind of a deal breaker at some point. So yeah, we're always looking for those next ideas, and we're a small team, and that's why we come to places like this to talk and get exposed to folks like you that might have some good ideas. So yes.
Are you familiar with the bioinformatics platform Bio4J? Specifically, my question would be, do you see any use cases for graphing some of the interrelationships between these things and doing, whether it's a community detection, cluster detection, or key brokers between populations, that kind of analysis?
Certainly, but I'll let.
Yeah, actually, we're all about exploring the graph relationships and actually using graph analytics to. One of the things that the genome paradigm has been dominated by for actually probably complicated conceptual reasons is this linear thought that everything proceeds in a linear fashion, and maybe it's because of chromosomal organization, who knows why. But what's emerging clearly is that biological phenotypes are network phenotypes. So we're actually all about trying to figure out how we can leverage more graph analytics and graph visualizations and other ways to get this data for sure.
Yep, fantastic. Yes?
Is your workloads are more memory intensive or really CPU intensive? Because the question is that it seems like yours is more memory intensive. As a result, your net CPU utilization will be pretty low on the process.
It is, and so that was one of the things that originally we attempted to do when we first tackled this problem, was to kind of right-size workloads to take the ones that were more computationally intensive and move them to that HPC workload where there's plenty of CPUs, and then take the memory-intensive stuff and move it over into the data-intensive environment. But currently, with that particular example is memory-intensive. So it's an interesting conversation because I talked to somebody a little bit earlier about the proposition of using, one of these high memory GPU pieces that has a ton of memory, but has a handful of cores, but they're only 1.1 gigahertz. This would be a good use case. Although we don't want to get into we don't want to build yet another appliance that only has a single usage, right? We want to build with flexibility. So and the timekeeper's giving me the sign. So thank you, everybody. Appreciate it.